Hello everyone, my name is Jiajun from the University of Tokyo. Today my topic is about biological modeling of feathers by morphogenesis simulation. Because our multidisciplinary research is highly related to biology, I will start from the summary of the biological background we have investigated. Feather is a special fur on bird skin. It likes human hair, but is way more complex. At the middle, it has a shaft, called rachis. There are two veins attached on this rachis, and we can split the vein like we did on the black feather in the picture. And we can see each vein consists of many fiber-like curves, called barbs, which is a core structure for representing the feather shape. On each barb, many barbules branch out from it. We can see these tiny structures more clearly on this green feather. They will form a locking system to link barb curves as a surface. Please notice the words in the boxes. They will be frequently mentioned in the following content. About the existing feather modeling methods for computer graphics, most of them choose the approximation way to define the feather geometry. The most practical approximation scheme will be the parametric definition. Each aspect of feather shape is defined by a parameter or a NURB curve, which is flexible enough to approximate feather shapes by user hands. Although in many cases, a real feather photograph is needed for reference. In film industry, artists may also choose to deform a NURB surface to feather shape, and then slice it to get the barb curves. In recent years, a more convenient way is to extract feather shape directly from real feather image, then generate barb curves using the old methods. However, few of them have considered what is the reason of feathers developing themselves into such shape. So, how feathers develop themselves? We can see the outline of different feathers can be smooth or can be sharp. Based on our observation, in most cases, we can find two relatively sharp points around the feather tips. It can be very sharp to make the feather tip no longer round, like the figure C. We start to think that this phenomenon may indicate there exists a common pattern among different types of feathers. And in fact, they are indeed from the same commonality. The result of the certain arrangement of barbs is decided by the skin cell development, which is the feather morphogenesis. Therefore, in the field of CG, we hope to provide a new feather modeling scheme by following the feather growth process. We do not try to defeat the approximation scheme, but to provide a new possibility to discuss how microscopic factors affect the macroscopic shapes of feathers. This correlation can further help us to discuss more difficult topics like feather physical simulation or feather pigmentation. We also hope to help biologists test their theories, assist their prediction, and become a tool for their data simulation. For feather morphogenesis, we will discuss three phenomena: the helical growth before feather maturation, and the unfurling and expansion after feather maturation. Based on the qualitative studies in biology field, we know that before a feather is mature, it develops inside a cylinder structure on bird skin, which is the feather follicle. At the bottom of a follicle, there is a ring of stem cells. They copy themselves and new cells move up. Then they will differentiate into barbs around the white circle area shown in the picture. If the cells simply move up and accumulate, the bulbs will grow straight down and no raches will be formed. This is how down feathers are developed. Note that this kind of feather has a more simple structure, so they are often rendered as fur in CG industry. However, if the cells shift a little bit while forming bulbs, they will grow helically and fuse themselves into a shaft. This is how counter feathers or flight feathers normally develop themselves. To elaborate the details, we show how the cross-section changes along the follicle. At the very beginning, bulbs have not been formed yet. Then, the first few of bulbs start to emerge along the circle. Then, two emergence positions at two sides become closer, and we can also see that bulbs are fusing towards the raches, like the bulb 1 here. Then, two emergence positions reach a red interval and stop become closer. After that, new bulbs only emerge from the red interval, and the existing bulbs continue growing towards the raches. And this process is the helical growth during feather morphogenesis. While the morphogenesis is proceeding, the older bulbs at the top of follicle are becoming mature and being pushed out from the broken follicle sheath. We can see the surface of the black feathers here automatically unfurl from helical state to a flat state. This is caused by the material of bulbs. This phenomenon is important to express the feather growth in 3D space, and also explain how a narrow follicle cylinder leads to a bigger feather. Furthermore, wide feather vanes look wider from the narrow follicle. When bulbs are released, because of their material, they will tend to expand and have a bigger angle to the raches. Therefore, vanes are becoming wider. But bulbs cannot expand freely. Barbules on each bulb will interlock each other, restricting the degree of this expansion. In biology field, the mechanical properties of bulbs is still under study, so we will focus on the geometric restriction from the barbules and see what we can achieve on simulating this restriction. So, we have understood some natural facts about feather development, helical growth, unfurling, and expansion. And in biology field, some people have attempted to establish mathematical models for reproducing helical growth in order to help their cellular or chemical studies. Plume and their co-workers firstly discussed the evolution of feather shape in 2001. They used 6 growth factors, like bulb angle or initial bulb count, to define the feather shape inside the follicle, which is essentially the same as the parametric definition method in CG industry, and it has already been highly developed. Then, Harris and their co-workers proposed an activator-inhibitor system to mimic the natural facts of helical growth. They used this system to mimic the amount change of real chemical substance during the morphogenesis, and the amount change becomes the traveling waves, which indicates the trails of helical growth. However, this system is aiming at bulb pattern and chemical substance, and it's impractical to generate bulbs to form a reasonable feather shape for CG usage. Furthermore, the complexity of this system can involve over 10 partial differential equations, with over 4 parameters for each equation, which makes it more impractical for keeping the whole system stable while adjusting those highly coupled parameters. In order to provide an easier adjustment while taking advantage of the feather morphogenesis, We propose a new way to reproduce helical growth by putting particles into user-defined velocity field. Besides, we also want to express the two phenomena after the feather is mature, the unfurling and the expansion. Although the material of barbs is still being studied, we haven't found any simulation studies for these two phenomena in biology field. So we propose our new method to reproduce them by using basic forward kinetics and barb snapping algorithm, and introduce our proposal into computer graphics world. Next, we will explain the concept of our proposal. For more details, please refer our paper. Before simulating the helical growth, we need to first build the mathematical model on the follicle cross-section. We treat the follicle cross-section as a closed cycle and is parameterized by the arc length. And in this one-dimensional curvilinear space, we need to define the Rake's interval and the Barb emergence interval. Each one is specified by a location pair on the cross-section. In order to simulate the Barb emergence, we define two particle emitters on the cross-section, so every particle will represent a new Barb. So, to reproduce the helical elongation of Barb, we let the particle emitters move inside a velocity field defined on the cross-section. Meanwhile, particles are emitted and start to move in a second velocity field. So, the trails of the particles, which is known as path line, will become the Barb curves. An example of this process will be like this. Whenever the emitter is delta d far away from the last particle, we generate the next particle at the position of the emitter. At the same time, all particles move in the opposite direction. Combining the upward movement, we can get the trails of the helical growth. About the calculation, because for any massless particle in a velocity field at a given time, the time derivative of its position must be equal to the velocity provided by the field at the current time and the current position. And the equation below is the integrated result for the emitter's one-dimensional curvilinear coordinate on the cross-section. We can get the similar formula result also for particles. In our implementation, we use Runge-Kutta fourth-order method to integrate the time derivative to get the position of the emitters or particles at given time. From the last stage, we can get sampled path line vertices as discrete curves in 3D space. And by the second velocity field, we can easily know the tangent at each vertex. Combining with the upward growing direction as binormal, we can build a discrete frame at each vertex. So, in order to apply the forward kinetics, we first transform them into chains by hierarchizing each vertex to the local space of its parent discrete frame. Then, we try to achieve the unfurling by doing the alignment as follows. Aligning every line segment to the tangent of parent frame, and aligning every tangent to the tangent of the parent frame. As for expansion, we first need to build the Barbules geometry data. Because the helical growth stage already provides the precise elongation direction of Barb, we choose to calculate the basis axis R for Barbules generation by using discrete frame. Then the normal vector, the basis axis R, and the vertex P can form a local hemispherical space. Then user can provide one azimuth angle and two zenith angles as the coordinates to define two Barbules vectors at each vertex. After having Barbules vectors, we can try to mimic this phenomenon by considering these facts. For a vane holding Barbules with hooklets, if the vanes are fully interlocked, each barb must fully snap to the adjacent one. Therefore, we first generate two groups of Barbules on one barb, the proximal group and the distal group. Then we calculate the boundary of the proximal Barbules, the blue ones. Then we generate Barbules for the lower barb. And then, for each distal Barbule, the red ones rotate current line segment to make this distal Barbule touch the boundary above. At last, we recursively apply this algorithm to each barb to simulate the geometry restriction of Barbules. This is a typical look of implementation of our proposal. We can see the overall shape of feather is mainly controlled by the velocity field for emitter and for particles. Here, we only need to define the magnitude at each position of these two velocity fields because their directions is mandatory by the morphogenesis. And the velocity fields are defined globally on the cross-section of the follicle. There is no need to separately adjust two vanes, but as a whole feather. There is a comparison of growth simulation result. In previous papers, we only found straights, and their coworker discussed the growth simulation, and their results were limited in 2D space and implemented by a simple interpolation of barb angles. Our scheme can achieve a 3D simulation of feather growth by applying helical growth and unfurling, which is more similar to the real developing feathers in the right picture. According to the biological studies, the Vane asymmetry on flight feathers is caused by the position bias of barb generative zone, the red interval. And in our scheme, by changing the red interval location, the shape changing of two vanes can be remarkably presented like the real flight feather and adjusted as a whole. Also, the turning point can be correctly presented by our scheme. By setting the velocity field smoothly for emitters, we can get the round tip, and by just giving a constant velocity field for emitters, the angular tip can be easily constructed. This kind of feather shape does exist in the nature. For example, the tail feather of white turkey. By applying our barb snapping algorithm, the pattern on vane can be more natural. Furthermore, by manually rotating one barb during our barb snapping algorithm, the deformation of vane surface can be simulated like the real feather. This also shows how microscopic information can affect the whole feather shape. In summary, we follow feathers' natural facts and propose a biological modeling scheme by mimicking three activities taking place during the feather development: the helical growth, the unfurling, and the expansion. And by our implementation, we find our scheme can positively reproduce similar biological characteristics on our output without particular modification. We also take the first step to discuss how microscopic information like barbules can affect the macroscopic feather shape, which is less studied in the field of computer graphics. But the disadvantage of our scheme is also obvious. We highly rely on the natural facts, but these facts have not been fully understood, especially the lax quantitative theories for further development. We have to follow the qualitative studies, which reduce some qualities of details in our output. In the future, we plan to investigate the effect of follicle cylinder size, because follicle size and rate of cell accumulation are actually changing during the morphogenesis. This is important for understanding how rachis is banded and how fetal variants are produced from the same kind of bird. Also, our Barb snapping algorithm needs to be improved. Since we have omitted the mechanical reason for Barb expansion, and our Barb snapping algorithm cannot reflect the material of barbs. It will be a challenging but promising direction to study the Barb expansion mechanism using bioengineering logic in the future. And the reference. Thank you for your listening.